So now we'll go back to the Earth in terms of the applications of AI, and the next three talks will be about applications in biomedical sciences. Our next speaker is Lior Pachter. Lior is the Bren Professor of Computational Biology and Computing and Mathematical Sciences at Caltech, and has many other distinctions, and I'll just let him take it over from here.
Thank you, George. I'd like to, first of all, thank George for organizing what's been a really interesting day and on a really interesting topic that haven't really participated in a meeting really focused on explainable AI like this. And I think it's fascinating and very timely, obviously, as many of the talks have shown. And I want to thank Kiri also for a really, really interesting talk. And I have to say it's very difficult to follow Mars, but I'll try my best. So I'm going to, as George said, focus a little bit on biology applications. And in biology, the feared response from Revere 3 is, yes, but what's the mechanism question, which gets right to the heart of interpretability. And especially in the current era of large-scale data collections, followed by applications of machine learning and AI methods, the question of what it means to be interpretable in the biology setting is central to the research that we do. And I'd like to talk a little bit about what that means, the how and why of interpretability in this context. So I'm going to start, though, with an analogy from geometry, of all things, which I actually think is going to be, in my view, a good metaphor for how biologists think about mechanism and interpretability. And I've put up a formula, which is that of the area of a circle in terms of its perimeter. I know it's after lunch and that part of the afternoon where the blood rushes out of the head, but hopefully everybody here is familiar with this formula and knows why it's true. But I want to ask you the question of, what would it mean to understand mechanism for this very simple formula? It sounds like a strange ask. And is there some sense in which one can speak of interpretability of this result? And I think there is, and I'm going to get to it. So the first thing I'll note, in case you really are knocked out without coffee, obviously this is true, and you could have done it in your head. This is something you learn in school, probably elementary school. The area of the circle is pi r squared, and the perimeter is 2 pi r. So you do the cancellations, and the area is the perimeter squared over 4 pi. But I'd like to note that when you do this calculation in your head, you don't really have maybe a sense, maybe you haven't thought of it for a while, about what is pi really. You may remember that pi is the ratio of the circumference to the radius, but if you really started to think about it, you would realize that that definition requires an understanding of what a circle is. It requires geometry, and it actually requires some digging deep into what you remember about, what you even know about what it means to really talk about pi. I'd like to offer you a definition that I like, and that is that pi is twice the smallest positive number at which the cosine function equals 0. That's because cosine pi divided by 2 is 0. But I'm going to take that as the definition. And now that I've done that, you might ask, well, what is cosine? And I'll remind you that that's, the unique solution to the differential equation, y double prime y equals 0. And we're buried in the weeds of calculus now in a talk about explainable AI for biology. You probably think I've lost my mind. This definition, by the way, is a good definition because it avoids the need for geometry. And in that sense, it's sort of, it is better in some sense, technical sense. It was popularized by the mathematician Edmund Landau. You may or may not know much about Landau, but you should know him in the context of this meeting because he is really the great-great-grandfather, you might say, of Google, especially the PageRank algorithm. he was the first to publish PageRank in 1895 in the context of ranking chess tournaments. Not quite PageRank, but centralized eigenvalues and essentially the same thing. And this was a Landaus preferred definition, so I'm not crazy for showing it to you. And lest you think that this is a silly matter about how we define pi and why it's related to interpretability, well, actually, Edward Landau lost his job with this definition as a pretext, because he was a Jew in Berlin before World War II, and the mathematician Ludwig Bieberbach, who was a Nazi, fired him ostensibly because he didn't use the right definition for pi, and he used the Jewish definition, which would be unsavory for Germans. And this is a text in German by Bieberbach about this. And he writes, vor eigenen Monaten haben Differenzen mit der Studentschaft den Lehrbetrieb des Herrn Landau in Ende bereitet. And I have the translation here in case you don't know German. He writes that, a few months ago, the differences about the definition of pi resulted in his termination because different races don't mix as students and teachers. So this matter of how you define pi, right, has been kind of sort of a big deal. Sort of derailed Edmund Landau's life, so to speak. And I mention it for good reason. So in terms of mechanism, this result about perimeter and this detour I went into Bieberbach, there's an actual result that lends insight, I think, into mechanism. And this is called the Isoparametric Inequality. That's due to Jakob Steiner, but a version of it called the Isodiametric Inequality is actually due to the same Ludwig Bieberbach, the Nazi I just referred to. And this isoparametric inequality says that the area of a circle, while it's equal to the perimeter squared over 4 pi, in fact, can satisfy, you can say something more, namely that of all shapes with fixed perimeter L, the circle has the largest area, and that's also true for shapes with fixed diameter D. The circle has the largest area. And in a way that is very much mechanism, because now you can think of a circle as, that's the shape where if the farmer wants to, build a fence and has a fixed amount of fencing, then literally the mechanism by which you will encircle the field will be a circle so as to maximize his crop area or her crop area. And so in a sense, this inequality, this theorem, which is in the form of inequality, provides the mechanistic insight for what a circle really is, including in biology, where really biology happens in 3D, so we think of a sphere, and lots of objects in biology take the form of a sphere, like the nucleus, probably for this very reason, that with a fixed amount of material, you can enclose the most amount of stuff. So what does this have to do with machine learning and interpretability? I'm going to get to that in a second, so let's store the isoparametric inequality, actually the isodiametric inequality of Bieberbach in the corner here. And let's talk for a second about dimension reduction and in particular variational autoencoders. So a variational autoencoder is, it's a parameterized form of an autoencoder that lets you have a generative model for your data. There's sort of two parts. The inference model, which is at the top here, whereby you have a neural network that takes as input your data and reduces its dimension. Then you have a second half to the model, a decoder, if you will. The first half is also called an encoder. The decoder lifts the points in low dimension, which are in the form Z, and raises them back into the ambient space in such a way that the points are as close as possible to the original data. And in an autoencoder, there's a parametric model on top of the latent space, which is a sort of technicality not important for what I want to say. And so with one of my former postdocs, Valentine Svensson, a few years ago, we thought a bit about the interpretability of such autoencoders. These models are not prima facie interpretable because the neural network has nonlinear functions. If you have linear functions, then you do have interpretability. Effectively, an autoencoder with linear functions becomes nothing other than PCA. And so when you do principal component analysis, you can absolutely interpret the latent space. Each of the coordinates is given in terms of loadings or linear combinations of the coordinates, if you will. But that's lost in an autoencoder. So what Valentine did is modify the decoder part, and hence we call this a linearly decoded variational autoencoder, or an LDVAE, whereby the encoding is nonlinear, but the decoding is linear. And this kind of mix and match of linear and nonlinear, it's a simple mod or simple modification to the neural network, comes at a cost of accuracy because indeed the linear restriction is very strong, or the linear constraint. But what you get from this is that you actually get interpretability on the actual coordinates, even though it's only half of the network. So in our paper, we showed that this is kind of a good, if you will, middle ground between a PCA and a nonlinear neural network, where you can interpret the directions in your latent space, even though you're benefiting from the nonlinear encoding on the first half. So, if you think about it in PCA, when you're projecting the points down into the latent space, the points in the latent space are, can then be the decoding part in PCA is sort of how you position that latent space back in the ambient space. And that's what we get from this linear second half. So it's a simple little modification, but it does provide some interpretability. And this is just an example from some data we looked at in our paper here on the bottom right. Now, what does that have to do with the circle that I started with? Well, there's actually a connection. So in recent work with my graduate student, Tara Chari, who's in the bioengineering department at Caltech, she produced a corollary or theorem in its own right, if you will, about two-dimensional embeddings. In particular, and it's derived from this isoparametric inequality. And what it says is that if you have a set of points in the plane, of course, you could also generalize this to higher dimensions, but we'll focus on the plane today for simplicity. And because that's also where people visualize and seek interpretability in their data. So in 2D, if you have a set of points, that doesn't really matter how you organize them. There's a bound on the ratio of the diameter to the minimum distance between the points. So one way to think about this is that if you only have 3 points, you can position them equidistant in an equilateral triangle. But of course, if you have 4 points, you cannot position them to be equidistant in the plane. You can do it in 3D, but not in the plane. And the more points you have, the more distortion you have to get between some pair of pairs. You're going to have some pair that are far apart and some pair that are close. And the significance of this is that if you have a high dimensional data set and you have equidistant points in high dimension, then it doesn't matter how you embed them in 2D, you will have a lot of distortion between these equidistant points in the embedding. And that gets to interpretability, and I'll say more about it in a minute, because if you're hoping to interpret something about your high dimensional data by looking at it in low dimension, and as Anima pointed out, this kind of visual interpretation is one of the 3 pillars or keys of general explainability in AI, you're going to have a problem that's just intrinsic to the geometry. And in order to prove this result, it's very simple, but it comes from this isoparametric inequality, because you can kind of fill up your space in such a way that you get a bound on the area. And this inequality, one sort of follows from the other. I won't do the proof here, especially not after lunch. So in our paper, Tara and I look at the sort of the implication of this sort of distortion for actual methods and actual data. And this is a plot from our paper, not produced with an autoencoder, although it could have been. It's produced with a method called UMAP, but we examined lots of different methods, and really all of them will suffer from this fundamental constraint imposed by this corollary. But what you can see here is we've done an analysis. This is from a biological data set. And before I talk about the picture, I should say what it is. It consists of single cell measurements from developing embryos in the mouse. It's from the group of Jacob Hanna, who's at the Weizmann Institute. It's from a recent paper that's exciting because it looked at a model by which development could be induced ex utero. And I'll show you in a second comparisons of ex utero and in utero data. But in any case, the data set here is a big matrix where for every cell in this developing embryo, there's been a measurement of gene expression, which means that there's a vector of approximately 20,000 different genes of how much their different abundances are in each cell. So there's a matrix of cells by genes, and these cells form groups, different types, and that's what you see in the legend here. We looked at one single type just to simplify things, chondrocytes and osteoblasts. That's why that's in the dashed form. And among these cells in this specific type, we looked for equidistant cells in high dimension. And it's interesting how you might go about searching for equidistant points, but I won't talk too much about that here. What you can see is that this particular dimensionality reduction technique has taken points that are fairly near each other in the ambient space, and it's positioned them in such a way that they're distorted. It's taken points that are actually far away but equidistant in the ambient space, distorted them as well, and you can actually see that it's also hard to see what was far versus what was near in the equidistant space to begin with. And that's the top versus the bottom. Now, this particular UMAP method doesn't lay a claim to trying to preserve the distances between the ambient space and the projected data. It claims its loss function is more attuned to preserving neighbors, but you can imagine that interpreting such a diagram, which people invariably do when they make them, is going to lead you to possibly wrong conclusions or misunderstandings about your data by virtue of just the naivete of looking at the picture and thinking that points that are nearby are actually nearby and points that are far away are actually far away. So we have a recent preprint about that, and I don't want to talk too much about it today, but I'll say that there's lots of implications to this fundamental barrier to embeddability and implications for interpretability. One of the ways in biology that these kinds of embeddings are used all the time to interpret are that you look at points from different batches or different experiments. Here, I'm showing in green and purple cells from embryos that have developed in utero versus cells that have developed ex utero. Now, it's amazing that we can actually develop an embryo ex utero, and that's the point of this really, really interesting paper. But the question in the paper in a quantitative sense is whether or not these embryos are really developing in the same way. And what we show in this figure here is that you cannot actually draw conclusions by looking at these kind of pictures, which is something that biologists do a lot. You can't do that because the embedding itself distorts, by distorting the relative distances, ends up distorting which points are near and which points are far away from each other. And you get, on the left hand, these are actually the same data, just normalized even in different ways. And on the left-hand side, the blue is the reality in the ambient space. We're measuring here the fraction of points of the X that are near the in utero and vice versa. So being 0 or 1 means you have lots of points that are the same. The points are separated. And the yellow is after the embedding, the points are mixed together, actually. So the embedding has made you think that the data is more mixed than it really is. And on the right-hand side, you see the opposite effect happening, just by virtue of applying the same method to data that's been normalized differently. So all of this requires great care. And the point here is that there are fundamental obstacles to interpretability, at least via visualization, which is, of course, not the only way that one interprets data. Final thing I'll say about this is Tara developed a neural net, which achieves something that I think is kind of interesting as a proof of principle, or if you like, as a thought experiment, which is that you can input a shape into her neural net and embed data into the shape. The upper left-hand corner is a map of the world, and the other three shapes are John von Neumann's elephant. This comes from a famous anecdote where John von Neumann quipped that with 4 parameters, you can draw an elephant, and with the 5th parameter, you can even get it to wiggle its trunk. And so this is sort of an anecdote about over-parameterization. And what we're trying to say here is that, in the top left, we've taken the ex utero data, and on the top right, it's the exact same data, and we've made it look like a map of the world. We've made it look like an elephant. And you can even see that the way we've done this, the points that are of the same cell types are clustered together. They actually have pretty good faithfulness to the distances in the ambient dimension, no worse than some other methods. But of course, the shape is arbitrary. So you shouldn't draw conclusions about how the data actually looks in 2D is kind of the point we're trying to make. And in the bottom half, you can see that actually totally different data sets. In one case, cells from the VMH region in the brain of a mouse. In another case, from a primary cortex, they can look like a mouse embryo. Really, they can all kind of look the same and have the same poor relationship to the actual distances in high dimension. It's just too difficult to go from 20,000 dimensions to 2 dimensions. But yet that's what a lot of people do. And the reason is that in biology, we are flush with data, and really a lot of the analysis of it is phenomenological and sort of just involves trying to find patterns in the data, draw conclusions about clusters in the data or other kinds of patterns. There's a sort of fundamental interpretability challenge in this sense. This is data from a paper that I wrote with my student Sina Boisagi recently. It's part of this paper and this research is part of a large consortium studying, in this case, the mouse brain. And this data is from the primary cortex of a mouse. That's what's shown in the top left with those arrows. And there are a lot of different kinds of cells and different cell types that have all sorts of acronyms. I won't get into those right now for this purpose. You can make one of these maps, as you see in the bottom left, but as I just said, it's really quite arbitrary. I could have had it look like a mouse if I'd wanted to. And the result that's gleaned from these kind of data is the kind of matrix that you see on the right-hand side, where we have different genes, or in this case, isoforms of genes. These are specific variants of genes, where we have markers for specific cell types telling us that this isoform of a gene is present in this kind of cell type and not in that one. And that's what people do with these kind of data. These little images on the left-hand side in the very first column in gray with black dots, those are actually spatial images of slices of the brain showing where in the slices these isoforms are actually present physically. So this is the kind of phenomenological analysis that's done, but it doesn't get to the question I got to in the initial, the beginning with the circle, which is mechanism, right? We have here sort of descriptive understanding of what was measured in the brain. We can do inference. We can run neural networks and try to spend a lot of time to interpret the weights of those networks, if we will, in the sense of typical explainable AI, in the way people think about what it means to be interpretable. But at the end of the day, you don't gain much insight into how the cell really works just by doing this kind of exercise. And in the biology domain, there's a lot of people that are troubled by this, actually. There's a very recent, this is from, I think, 2 weeks ago, or even last week, I guess, opinion piece by the famous biologist Paul Nurse in the journal Nature, which is basically, it's a short opinion piece without much more content than this title, which is to say that, yeah, these data are great, but just clustering the data or looking at them very phenomenological like this doesn't actually give insight into mechanism. What's really missing is the generation of ideas and methods to get at those ideas. And so in my group with students of mine, Gennady Goreng is from chemical engineering, and Meishan Fang, who's also a graduate student at Caltech, we've been thinking a lot about how we could go about developing mechanistic models from the kind of data that are being generated in a way where we can have really true biological interpretability. And so in a collaboration with another graduate student from Vanderbilt, John Vestola, we've recently written a paper that considers interpretability to be a process rather than a specific output from an inference procedure. And by process, I mean that really the interpretability from the inference goes hand in hand with designing and then executing new experiments. So what I mean by this is we considered a setting where we might want to examine two different models, and by this I mean biophysical models that could be generating the kind of data that we're obtaining about measurements of gene expression in cells. We looked at a mathematical analysis of a pair of models, I'll mention in a second what they are, to try to understand what kind of information would allow us to distinguish between them. That in turn can go into thinking about design of experiments that would yield the data that would allow for distinguishing such models. And then in theory, one could rule out one model or another and go back to the drawing board for more refined discrimination between models. So the models we considered were two classical models from actually the finance industry. These are stochastic differential equation models where we have a model for the rate of transcription over time, sort of reverting to a mean and having an addition of stochastic noise. And these models are called the Gamma-Ornstein-Uhlenbeck and the Cox-Ingersoll-Ross model. We picked these models because we had some motivating biological connection or explanation for them that we felt that was physically interesting. But of course, you could consider all sorts of models. And what we're sort of proposing to do in this paper is to study these models to understand what experimental data it would take to distinguish them from each other. I won't talk too much about these results now, especially because I'm nearing the end of my time. And I know we're a little early, so I'll speak a little bit longer, but I wanted to be within half an hour. Each of these kind of models has a different implication for the kind of noise it generates and the kind of data you will measure in these kind of experiments. And so, to summarize it, we feel that this is really the key to sort of, in this domain at least, to providing interpretation to the sort of data we're collecting and to the models that we're using for them, is to not just use neural networks for inference and prediction, but to couple them with biophysical models that really directly model the actual biophysical parameters of interest. So I'm gonna end with that, and I will thank many of my students in my lab and collaborators. So some of these individuals are collaborators of mine at other institutions, and I've highlighted the people that specifically contributed to the papers I've mentioned, but all of these are students and collaborators with whom we've been discussing exactly these issues of interpretability and mechanism for this domain for a long time. So I'll stop there and see if I can answer any questions.
Thank you, Lior. That was great. I believe there is one question so far, which is just wondering how you measure the distances. Let's say if we just measure the expression of one gene and we get this expression in 3 cells with counts 1, 100, and 1000. Do you think the cell 2 is closer to 1 than to 3?
Okay, that's a great question about distance. So first of all, there's a complement of about 20,000 genes in a cell. And so the question in a mathematical form is, how are you measuring distance between two such abundance vectors? That's a great question. In this domain, first of all, I'll tell you what generally people do is they use the regular Euclidean distance or L2 norm. Some people in this particular, for the paper I showed you the results from, we used the L1 norm. L1 is generally preferable in very high dimension because all points become look equidistant in a sense when you use Euclidean distance in high dimension. So we prefer L1 norm. Having said that, it is very challenging. You know, there's a challenge in thinking about what distance ought to be and what appropriate metrics are. We've discussed a lot in my group, and I know others have thought about this as well. You know, what kinds of metrics make sense? And it's just exploring that question is a whole project that really I don't think has been addressed satisfactorily, at least for me, in this domain of single cell analysis. And I should also point out that gene expression is just one kind of data or modality that can be measured these days. There's many others, and there's presumably different metrics that make sense. So understanding what a good and correct biological metric is a really great question.